Pues muy bienvenidos a la plenaria. Vamos a dar unos minutos para que terminen de acomodarse. Voy a empezar en estos minutos, mientras terminan de llenar el salón, agradeciéndole muchísimo a Jacinta Conrad, que aceptó venir a dar esta plática. Ella vino a una reunión en Guanajuato sobre materiales fuera del equilibrio y aceptó en su regreso a su casa pasar a vernos. Y bueno, me acaba de decir lo contenta que está de conocer la Academia Mexicana de Ciencias y de haber podido hacer este pequeño alto en su camino. Me preguntaba que cada cuánto hacemos este tipo de cosas, le dije que era la segunda, y bueno, le parece fantástico que la Academia Mexicana de Ciencias organice una reunión de este tipo. Les voy a decir un poco sobre Jacinta Conrad. Ella es Associate Professor of Chemical Engineering y Associate Professor of Petroleum Engineering at the University of Houston. Recibió su licenciatura en matemáticas de la Universidad de Chicago, su maestría y su doctorado de la Universidad de Harvard. Después ha trabajado primero como un postdoctorado en ciencia de materiales en la Universidad de Illinois, antes de irse a Houston en 2010, donde ella está trabajando en algo que nos va a contar en este momento, en el transporte fuera de equilibrio, en fluidos complejos y su aplicación en el diagnóstico, los prototipos y las superficies que tienen necesidad de este tipo de tratamiento. Ahí tienen el título de la plática. Yo, sin más, le cedo la palabra a Jacinta. Thank you so much, Jacinta, for being with us. And please.
Gracias por la invitación. So today I'll talk about working at the boundary between physics and engineering. My training is in physics, but because this is for a broad audience, I wanted to focus on some of the work that we look at that has implications for social growth. And so what I'm focusing on is looking at how the non-equilibrium science that we do and that other scientists do can start to contribute towards large-scale challenges for society. So what are those large-scale challenges? Very briefly, we're interested in being able to provide good health for citizens, so food and medical care, access to clean water for drinking, provide sufficient energy to meet society's rising demands, and in each of these challenges, be able to do this in a sustainable fashion, in a way that will allow citizens to be able to continue to grow and prosper across many generations. So towards this end, thank you, technical challenges. Towards this end, the United States National Academy of Engineering has identified 14 grand challenges for engineers. These include advancing health informatics, being able to manage the increasing amount of data that is expected to be collected by new techniques to diagnose and treat medicine. Similarly, to engineer better medicines, to treat conditions that are so far inaccessible to treatment, to provide access to clean water, and to restore and improve urban infrastructure. So these are big problems. And what I want to talk about is how non-equilibrium science and how developing fundamental understanding can start to contribute towards making small progress on these very large-scale challenges. So I first want to introduce the concept of equilibrium. Equilibrium can be thought of in two ways. Scientifically, equilibrium is a state of motion and internal energy that is steady over time. So on the left, I show an example of mechanical equilibrium. This is a state in which the rocks are perfectly balanced and nothing is moving. On the right, I show an example of thermal equilibrium. So in cartoons, a mechanical equilibrium occurs when forces in a system are balanced. So for example, if I set this clicker on the table, the clicker pushes down with the force of gravity. The table pushes up against the weight of the clicker, and everything remains static and stationary. Similarly, for thermodynamic equilibrium, here I show an example of gas equilibrium, in which a gas expands to fill the volume of its container. Many of the achievements of 19th and 20th century physics have focused on understanding the equilibrium properties of matter. Nonetheless, the challenge for the 21st century is to start to explore and understand the non-equilibrium properties. So first I give a couple examples in which the ways in which processes or materials can behave out of equilibrium. So for example, it's well known that friction, a mechanical force or a mechanical dissipation, can be used to convert mechanical motion into heat and start a fire. Similarly, acceleration can change the direction of motion or the speed of an object. These are non-steady state processes. Similarly, systems themselves can be in non-steady state. For example, in school, we learn that materials are either gases, liquids, or solids. A gas is a container, is a suspension of non-constant volume. A liquid has constant volume, but both gases and liquids conform to the shape of their container, whereas a crystalline solid has atoms that are arranged in a regular lattice. So we can look at the atoms and understand the equilibrium properties of these materials. More recently, though, scientists and physicists have started to explore the properties of systems that are out of equilibrium, that are arrived at through different formulations. So, for example, we can look at systems that contain stable clusters in fluid that rearrange but never fully aggregate or arrest. Or mechanical gels. These are materials that we use in everyday life. Or mechanical glasses. And again, these can be distinguished by the arrangement of their atoms as well as their evolution over time. For example, the glass is structurally indistinguishable from a liquid, yet it behaves mechanically like a solid and does not flow. So what then is non-equilibrium? These are systems in which the state of motion and the internal energy are not steady over time. Okay, looks like it's going to stay there. Great. So my system itself was not in equilibrium. Okay, so what is non-equilibrium? These are processes that may depend in space or on time. They can be exposed to driving forces such as gravity or flow. The systems themselves can be heterogeneous. Different portions of a system can look different, or the system can evolve in space or in time. Similarly, non-equilibrium processes may use or dissipate energy. And so I showed an example of friction, but in general, chemical reactions and the activity underlying life itself are all examples of non-equilibrium processes. So I want to focus on three types of non-equilibrium behavior. The first of these is the simplest. This is a case of flow. Here I show a movie of a flowing stream. This is a non-equilibrium system. You can see that the velocity, even on average, of the water as it flows varies in space and in time. It depends upon interactions of the fluid with the boundary of the system. For example, the grasses along the stream edge. And there are significant fluctuations in the flow properties over time. Although we normally think about liquids flowing, we can also have flow even in highly non-equilibrium solids. This is a movie of a polymeric solution called pitch. I have you look at the time scale in the corner. So a day goes by every few seconds. This liquid is so slow that this experiment has been running for 80 years. And 8 drops of liquid have fallen in 80 years. Okay? This is a material that has a viscosity, a resistance to flow, that is 230 million times that of water. Okay? So this is an example of a non-equilibrium system subjected to non-equilibrium forces. What's the opposite of flow? Okay. Well, the opposite of flow is arrest. And I hear you have problems with this kind of arrest in Mexico as well. Okay. So this is an example from China. It's a 50-lane traffic jam. Okay. And all of these cars, I'm going to pause for 1 second. All of these cars are returning from vacation, and they're all trying to go through this very, very narrow gap here. Okay. 50 lanes. Okay. And so you can see that systems can arrest when subjected to heavy constraints or to high densities. This is an example of a moving system driven by people, literally. But inorganic systems, artificial systems, can also arrest and jam. So here I show a movie from Bob Berenger's group at Duke, where they look at the flow of model grains through a very narrow orifice. Just as in the case of the cars, eventually a structure forms at the narrow orifice that prevents anything from moving. And this is remarkable. This is a system that flows like a liquid and then becomes arrested like a solid. So the final example that I want to give of the ways in which we think about non-equilibrium motion is a case of motion itself. During the coffee break, to get energy for my talk, I went and had 3 cookies. Okay? And indeed, my activity as a human is driven by the consumption of energy in the form of food. But all living organisms exhibit activity. And so here is an example, not of people, but of bacteria, whose size is approximately 1/1,000,000th of a meter. And they're sitting on a surface, they consume energy, and you can see that they exhibit a wide variety and diversity of motion. Okay? And again, this is a non-equilibrium process because they must continually use energy to be able to move. So non-equilibrium is really the behavior of life itself. And inspired by life, sitting with my engineering hat, many engineers now are interested in being able to think about designing materials that mimic natural properties. So for example, here is a movie of a flower that slowly closes over the course of a day. Engineers have now started to understand material properties so that they can create artificial analogs of these kinds of materials. So here I show 2 examples of polymers that, when subjected to a stimulus, either water, by immersing a polymer in water on the left, or by irradiating the polymer on the right, are able to fold up in the same way that the flower closed. So this is the focus of engineering, but really at heart, I'm a scientist. So what is the role then for non-equilibrium science as we move forward? So the role, as I see, for non-equilibrium science is to understand physical principles that underlie these amazing responses and changes in properties over time. For example, being able to understand how systems respond to forces such as gravity or flow that may disrupt them, to understand consequences of the fact that these systems are indeed heterogeneous, and to understand the consequences of properties that change over time. So for example, if you were sitting in the non-equilibrium symposium this morning, you heard about examples of glasses. These are materials that are solids that nonetheless exhibit remarkable changes in properties called aging. And you heard about colloidal gels. These are materials that are heterogeneous. Similarly, non-equilibrium science seeks to understand the consequences of energy use and dissipation. And in particular, much of non-equilibrium physics now is interested in the results of reactions or activity that drive natural motion. So that's my introduction to what non-equilibrium science is. In the remainder of my talk, I want to focus on work that my lab does, but I want to motivate it through this framework of trying to address large-scale problems. So my lab's work is focused on looking at confined flow of complex fluids. Complex fluids, again, as we heard this morning, are liquids that contain small particles. And you can see, for example, the blue dots that flash through these pictures over time. These can be nanoparticles, whose size is 1/billionth that of a meter, or colloids, 1/millionth, or other kinds of dissolved constituents, such as polymers or surfactants, the active ingredients in soaps. So we study these systems in many cases as they're being flowed. So here I give one example in which we're flowing nanoparticles through packed beds. And these are very simple models, physical models, of processes to try to understand how particles spread out as you go through a porous medium. So why do we care? These are very simplified models, for example, of the ability to deliver drugs throughout the body formulated as nanoparticles, or the ability to deliver nanoparticles that act as viscosifiers or gellants to enhance and increase oil recovery. So as scientists, my group does lots of imaging. So we take movies like the one that I'm showing, and we use methods that allow us to follow the trajectories of all of the particles over time. Here, all of the trajectories are color-coded by the speed at which the particles move. And from this kind of behavior, we're able to extract out properties that characterize the transport of these different dissolved species. So this is science, but now I want to start with thinking about how we address large-scale challenges. So I'm going to talk about 3 areas in which I think our work is relevant for the grand challenges that I identified in the beginning of my talk. So the first 2 challenges that I want to focus on are advancing health informatics and engineering better medicines. These are really worthwhile challenges, right, to improve human health. Both of the descriptions of these challenges identify as a critical need the ability to improve diagnosis. These are methods that allow medical conditions to be identified. In particular, I came at the beginning of the week to attend the wonderful meeting organized by Ramon on non-equilibrium systems, matter out of equilibrium, and I flew to Mexico on an airplane. As we gain ability to move around the globe, one of the challenges is in combating viral outbreaks. So for example, in 2014, the outbreak of Ebola, especially in Western Africa, created enormous challenges for treatment there, but also for treatment for visitors from these locations. These viral outbreaks often appear in places where there are limited scientific resources. Similarly, more recently, we've all heard about the emergence of Zika virus, which threatens my hometown of Houston, Texas. That's the dark red dot. So this is an emerging challenge that is exacerbated by the ability of people to spread rapidly around the globe. Hence, treatment methods demand needs to diagnose viral outbreaks. And these methods must first be sensitive to detect only a few viruses, as well as simple to be able to use in settings that are resource limited. Why is that important? Well, most existing diagnostic methods are lab-based. They occur in large hospitals or commercial laboratories. They use equipment that is sophisticated and very expensive, and they require trained personnel. By contrast, one appealing route to thinking about diagnosing and detecting disease is to use diagnostics that are point of care, as shown by the glucose meter here. These methods can be used in clinics, in places lacking laboratory infrastructure, or in settings where privacy is demanded, such as testing in the home. These methods are low cost and simple, and require few or no trained personnel. So we got interested working with my collaborator, Richard Wilson, to look at trying to understand how we can improve a common point of care diagnostic format. This is called a lateral flow assay. So why might you know about a lateral flow assay? This is the method that is commonly used to detect pregnancy. So in a pregnancy test, samples from a woman who would like to determine if she is pregnant, typically urine, are placed at one end of this strip, and they're transported through a porous membrane that sits within the test. These work via what are known as a sandwich assay, something that will generate a visual signal. So the blue color here binds to a particular protein in a woman's urine and then binds to the porous membrane to generate an easily read visual signal. So here, the control line indicates that the test works correctly, and the test line indicates that you need to plan better 9 months from now. Okay. So this is a great format, okay? I don't know the cost in Mexico, but this format of test in the US costs about 13 American dollars. And if you want, you can buy very cheap strips without the plastic housing for about 10 cents on Amazon. Okay. So this is cheap, disposable, and point of care. Why aren't these formats more widely used? There's one significant drawback. They're insensitive and they're not quantitative. This is okay if you're diagnosing pregnancy. You're not a little pregnant. You're pregnant or you're not. Okay. But if you're looking to halt disease progression, then understanding how many of a virus is important for determining treatment. And the numbers that are relevant for viral outbreaks are quite small. So for example, Ebola has between 1,000 and 1 million viruses per milliliter of a sample. And the dengue virus, similar to Zika, has between 10 and 1,000. So developing methods that improve the quantitative ability of these tests is critical for their applications to detect disease. So how does this test work? I described it, but I'll go back. Here, inside these tests, contain nanoparticles, particles that are 1/1000th the size of a meter. And they're functionalized with antibodies to proteins that appear in the urine. And so as the sample is transported by capillary flow through the strip, the reporter particles bind to the proteins and are turned captured on the test line on the strip. And so with our colleague, we showed that we can start to focus on replacing two of the features in this assay to improve sensitivity. And specifically, we highlighted first the reporter particles. So we replaced nanoparticles now with model viruses. These viruses are well established as materials for nanoscale engineering. These are M13. They're a virus that infect bacteria, but not humans. But because scientists have a wide range of abilities in being able to functionalize these, they can be conjugated with elements that make them critical for recognition. So you can add something on their tip that allows them to be able to bind to the analyte of interest. And you can also add fluorescence groups that enable them to be imaged with microscopy. Okay. These are rods, so they're 1 micron long and only about 6 nanometers wide. My group loves looking at things, and so we replaced the visual line in the pregnancy test with microscale methods that allow us to count individual reporters. And with these two innovations, we showed that we were able to get a test result, I'm showing here, that was 100 times that of a comparable LFA. Okay. So that's great, 100-fold improvement in magnitude. So now you might be saying, so far you're telling me about engineering, where's the science? So this was all worked great. What didn't work great? We didn't understand why. And so my lab specializes in understanding how to control the flow of materials through complex media. And we hypothesized that something about how our improved reporters were flowing led to this advance in sensitivity. So here I show you a schematic of the model device that we use to image reporters as they're transported. We make a very simple model of a lateral flow assay. We obtain only the porous membrane strip. We put our microscope underneath the strip, and then we add a drop of fluid that contains our reporters. We then watch as the reporters are captured at the test line, which is functionalized with chemical groups that allow us to capture the reporters. Here is just a camera picture of our experimental setup. And here then is a picture of the membrane itself. So again, we love to look at things. So here I show you a movie in real time as the reporters are transported through the strip. The fibers of the membrane are shown as long rods, and the bright dots are the reporters that we use for detection. Okay. So we analyze this using the tracking and analysis methods that I highlighted on my previous slide, but I'm going to give you the qualitative picture of what we learn from our science. So the first thing that we learn is we carefully track the position and orientation of each of the reporters as it approaches one of these porous membrane capture points. And what we see is that as the reporters come in to be captured to a rest on the membrane, that they often undergo rotation. So why is that important? So they tumble end over end. And if you remember, my reporter had a capture group on one end. And so by looking then at the processes that drive capture, and by looking, I mean detailed tracking of the particles coupled with numerical simulations of the flow field around the membrane, we're able to show that the rotation is a critical aspect that improves capture. If you like, rotation drives the ability of the recognition element on our reporter to be captured on the strip. Okay? And this helps to promote binding because the sticky end then is more likely to contact with that strip. So this is what we learn from doing pore scale imaging in these systems. How do we translate that knowledge? Well, if rotation is so important, then how do we make more sensitive reporters? If we make them longer, does that improve their ability to be captured? So we worked with our collaborators to come up with a list of different reporters of widely varying length and aspect ratio, and we made them into model reporters. And what I want to show, we did a series of images. This is the only graph I'll show here, but I just want to say that for the longest reporter, shown in the blue symbols here, that exhibited the greatest likelihood of capture. And so that's important. We've taken fundamental insights from fluid mechanics, namely that particles will rotate when subjected to a vorticity field generated by the gradient of velocity near our membrane. And we've used that then to understand how we change the ability of our reporter particles to stick and be captured. We're now exploring the relevance of this for detection in a realistic setup. So that's the first vignette I want to tell you. I wanted to talk about non-equilibrium science for diagnostics. What I've shown is that the sensitivity of a common format for point-of-care diagnostics, the lateral flow assay, can be increased using viral nanoparticles. But using science, we understand that that increase comes at least in part from the non-equilibrium transport properties of our reporters as they flow through the membrane. And this is an advance that's not possible without collaboration with chemists and with engineers, and also without discussion. So this we expect will have impact. More sensitive methods to detect and diagnose disease allow disease to be detected earlier in progression. And that's important. That can prevent the spread of viral outbreaks. And so this may help to reduce pandemics and/or increase survival chances. Finally, I want to emphasize, I'm talking about science, but the large-scale challenges that I've identified are not challenges for science alone. And so indeed, there are social, political, and legal challenges that also must be addressed. Because this is an interdisciplinary meeting, I'd like to highlight some of those. So to use these kinds of ideas to advance health informatics requires the ability to collect and maintain data between health agencies. That's a logistics challenge, but it's also a political one, as viral outbreaks know no borders. To engineer better medicines and diagnostics means that we have to be able to manage the data that are generated by the diagnostics over time. So the second focus area that I want to address in the context of grand challenges is the need to reduce fouling by bacteria. So two more of the NAE grand challenges, that is providing access to clean water and restoring and improving urban infrastructure, identify as a critical need the need to reduce biofouling, that is fouling of surfaces by bacteria. And this need in turn motivates new strategies to coat materials. So what kinds of surfaces will bacteria stick to? The answer is everything and anything. Okay. I hope, I love talking about toothbrushing in this context. This morning before you left the house, why did you brush your teeth? So you know that it's to prevent tooth decay. And so what causes tooth decay are bacteria that you ingest that then stick to your teeth, and we'll talk about the kinds of communities that they form. Bacteria are extraordinarily clever. They can colonize any surface. So for example, I show examples here of fouled pipelines, of fouled water treatment membranes, and fouled materials for infrastructure. And the damage on these materials is all due to the presence of surface-associated communities called biofilms. So it's long been thought, and there's been much interest in understanding bacteria in the planktonic state as they freely swim in fluids. But as the Nobel laureate Walter Brattain noticed, much of biology is really concerned with reactions at a surface. So the micrograph on bottom shows a picture of a bacterial biofilm. Over 99% of bacteria live in these surface-attached communities. And the reason that they form biofilms is the reason you brush your teeth, that the communities that they form are extraordinarily resistant to stresses and strains. Extraordinary. They can withstand tremendous mechanical forces. And hence, there's a growing interest among biophysicists and material scientists and engineers in understanding why and how these communities form. So in the typical paradigm for biofilm formation, biofilms form when bacteria first come down to a surface and first initially and then irreversibly attach. Upon irreversible attachment, bacteria then start to secrete polymers that create the house in which the bacteria live. And these polymer substances protect and shelter the growing community until finally the community becomes large enough so that cells are able to redisperse. So we're interested in trying to prevent how bacteria stick. We want to do that physically. So why is this a problem for physicists? Indeed, biologists have been watching things stick to surfaces, but typically they count cells, and they simply count the number of cells that appear over time. Okay. But this is a physical question because this process is active and requires dynamics. So here I show a movie from my lab in which we watch bacteria deposit on a surface. And you can see that, first of all, they come in and sometimes stick, they sometimes unstick. They don't always come in and attach irreversibly. Sometimes they can move while sitting on the surface. So this process is dynamic, this process is non-equilibrium, and this process involves motion. So from the standpoint of non-equilibrium science, we were interested in understanding what this kind of mobility near the surface could tell us about the interactions between bacteria and surfaces. So again, we designed an experiment that allows us to access bacteria as they stick to surfaces. This is a schematic of our microfluidic flow cell, and we watch bacteria attach over time. We use a model bacterium. This is E. coli, what makes you sick, but it also, these strains are non-pathogenic. And this is a nice model system because, as always, bacteria are complex colloids. Okay? They're microscale particles, but they have surface structures that help them stick to surfaces. And these include appendages known as fimbriae, which roughly act like Velcro, and flagella, which act like rotary motors and allow the bacterium to swim. And you can see that this adhesion process is indeed highly dynamic. So to gain insight, we looked at adhesion of bacteria on a wide range of surfaces. We used microscopy methods and our eyes to try to understand what are the differences in properties. So I contrast here a less sticky surface on top and a stickier surface on bottom. And if you contrast the behavior of bacteria in the two movies, the very sticky surface, nothing moves. Bacteria come down and stick irreversibly. Whereas on the less sticky surface, there's a lot of wiggling. Their cells will attach, they will detach, and they'll move along. So we thought that by analyzing the motion using the same kinds of imaging and tracking methods that we used for particles and that we used for viruses in the previous diagnostic, we could gain information on surface interactions. And so the way we do that is we quantify the number of bacteria that move a lot. And what we can show is that over a wide range of surface treatments, as well as a range of flow rates, that the likelihood that bacteria move, shown on the y-axis, is inversely correlated with the rate at which they deposit. Okay? Or more simply, the more bacteria move, the less likely they are to stick. We similarly did another set of experiments probing the relationship between surface properties and motion. For some surfaces, we noted that bacteria would actually spend a large period, that bacteria could be entrained along the surface for long periods of time. So the cell that I circle here is moving very slowly along the surface. It has no appendages that drive this motion. Okay. This is a hydrodynamic motion, if you like. And on the right, I show trajectories. How do we gain insight? We again use a combination of chemical methods to pattern surfaces and physical methods to help us analyze their motion. And what we can show is that for bacteria that lack the sticky Velcro on their surface, these are the bacteria that undergo this mobile adhesion, but they only will do this when the surface itself is very, very smooth. Okay. And so here I show two surfaces that are almost identical in their chemical functionality, but are very different in their surface roughness, as shown by the height plots. Okay. So our studies then of the non-equilibrium behavior of bacteria near surfaces suggest that motion is inversely related to detachment. And so we wanted to use these ideas then to suggest strategies to prevent bacteria from fouling. So similar ideas for using bio-inspired approaches to prevent fouling are widely known. So for example, the membranes of eukaryotic cells bear both positive and negative charges. And so similar artificial zwitterionic positive-negative coatings have been shown to reduce fouling by bacteria. Okay. Similarly, the skin of sharks that swim in the ocean has a series of regular microscale ridges. And so engineers have used this idea to create natural anti-fouling topographies. Our studies from science suggested that motion was key. If we could encourage bacteria to move, we might be able to detach them from the surface. And this is inspired by natural creatures such as the mollusk shown here, okay, which have structures that are known as cilia on the outside. And you have cilia too inside your nasal passages. And the purpose of both of these is to move and to clear away any kind of foulants. So inspired by this approach and inspired by the idea that we knew that motion was critical for being able to prevent fouling, we're working with a colleague, Megan Robertson, who's a polymer chemist, to now exploit non-equilibrium material properties to start to push bacteria off the surfaces by making them move. So I'll just show one set of results. We do this using surface-attached polymer brushes. And these are charged when the solution is at very high pH, when it's very alkaline. And so here, the brush is swollen and extended, whereas in acidic solutions, the brush is uncharged and collapses and repels water. And so by measuring the angle that water makes on the surface as we switch the pH, we can show that we get reversibly switchable surfaces, and moreover that the extent of the switchability depends on the properties that we program into our material. And so we're now in the process of exploring the use of these materials as anti-fouling substrates, with the hypothesis that moving the surface will detach the bacteria that themselves are mobile. Yes, and so we can actually do this reversibly and dynamically. So yeah, we're hoping it's going to work. It's sort of working right now. That's why you get to see this. Okay, so yeah, this is a very common strategy that again mimics natural properties. So what have we learned from our non-equilibrium science? Again, we've shown that near surface motion of bacteria not only probes surface properties, but surfaces that promote motion exhibit reduced deposition. And so we're using this as a way to think about how to design self-cleaning dynamic surfaces that detach loosely adherent or mobile bacteria. And we hope that these self-cleaning surfaces will reduce fouling. And that will in turn help us to reduce damages and costs on some of the most commonly fouled surfaces. Again, this is a social challenge as well as a scientific or technical one. The need to promote clean water is not just technological. Places that lack fresh water have to be encouraged to reuse water through filtration membranes. That's a social challenge. Similarly, in the United States right now, we have a $2 trillion infrastructure deficit. $2 trillion of deferred maintenance on roads and bridges and pipelines. That's a political challenge because it's easier to ignore the problem until it's too late. So the final challenge I want to talk about is one that really is not directly related, but underlies many of the NIE grant challenges. And I've highlighted some, but I'll particularly focus on this last one, engineering the tools of scientific discovery. Really, all of these challenges require the needs to precisely control structure and materials over tremendously large length scales. And so I'll focus on techniques that control from the nanometer scale, or 1/1000th of a meter, up to the sort of centimeter to meter scale. And I'm going to focus on a particularly popular technique. Right? Everyone's heard of 3D printing, right? A lot of excitement. And so 3D printing is amazing, right? So here is an example of beautiful 3D printed structures. Okay. Beautiful, not yet so useful. Okay? And so indeed, most current 3D printers work by extruding plastic resins. Whereas if we want to think about building useful structures on demand, we need to think about materials that can do something. Okay. So what do I mean by that? Well, for example, what if I want to print a three-dimensional antenna? Okay. Or what if I want to three-dimensionally print a house? And these are both actual examples of 3D printed materials. And so going from beautiful toys to functional materials requires that we think about how to incorporate that functionality. So I'll focus in, even more for our science on a family of techniques known as direct ink writing, which I show here as a movie. It's going to come back. So through these techniques, a concentrated ink, where did that go, is extruded, and you can draw out structures in a layer by layer fashion. And so here's the movie, it's looping again. And so using this technique, pioneered by Jennifer Lewis at Harvard, they've shown that you can build amazing functional structures. So for example, if they design an ink that contains silver nanoparticles that allows the material to be conductive, that they can draw out gap-spanning wires and then print microelectronics. Similarly, they've more recently incorporated silicon carbide whiskers and carbon fibers, and these provide mechanical reinforcement to create structures with the amazing strength and lightness of balsa wood. Okay. So now, what is the challenge if we think about controlling particles in flow? Again, I do love talking about toothpaste. I'm a soft matter physicist. So this is an example of a material that has been engineered to control both flow and solidification. When you push the toothpaste out of the tube in the morning, it has to flow like a liquid. Once it's on your toothbrush, though, it needs to remain there and not fall over your bedside, over the sink. And so this is really an amazing engineered property. This material has to flow like a liquid under certain strains, and then solidify, form a solid when quiescent. And so as an engineered material, this and other personal care products have been highly engineered for their non-equilibrium flow properties. And indeed, the flow properties of these materials are carefully tuned to be controlled across the range of relevant extrusion rates. So the role then for non-equilibrium science for processes that involve flowing particles through very narrow channels, okay, and simultaneously preventing clogging, like we saw in the Hopper movie, is to think about tuning the interactions between the microscale constituents. So for 3D printing, a convenient feedstock are colloidal gels, which was discussed this morning by Ramon. These are particles that have interactions that make them sticky, and so they form space-spanning structures. The particles are on the size scale of 1 micron, and they have to be able to flow through geometries that are between 1 and 100 microns. But as soon as they flow and they're extruded in that printing process, they need to be able to re-solidify. And so my group has been interested in trying to understand the microscale fluid mechanics of suspensions that must behave like solids and liquids. Again, we design microfluidic devices, and we tune the interactions between our particles so that we get weak repulsion or strong attractions, or anything in between. And you can see from the movies that I show here that these have very different properties, that the strong attractive particles do not yield into individual particles but form clusters, whereas the weak attractive particles just flow as individual particles. Why is this important? Well, by tuning interactions, we can tune the likelihood that these materials flow steadily without clogging or jamming. So from the same kinds of single particle tracking and analysis techniques, we can calculate the density profile as these solutions flow through the channels, as if all along the channel. And in the case of weak repulsions, we find that the normalized concentration steadily increases downstream, that our suspension is trying to clog itself. And this is because fundamental fluid stresses push the particles towards the center of the channel, and the suspension lacks a compressive modulus to prevent it from densifying. Once we introduce an attraction, we maintain uniform density. And this is important for a printing process. And this also maintains conductivity, which is important for functional properties, such as conductivity. So the final vignette, I told you that tuning interactions between particles flowing in a fluid modifies the density profile. And we can use this then to keep the density profile constant during flow to prevent clogging or jamming. And these kinds of understanding the relationship between flow and particle attractions is important for imparting function to printed parts. And this function can include optical, conductive, or mechanical properties. And the reason why people are so excited about 3D printing is it allows you to make materials on demand. And this is a benefit for sustainability because then you can only create the parts that you need. And again, finally, this still requires that access to the methods be granted to the wider public, and that's a social challenge. Okay, so very quickly, because I'm slightly over, I've left highlighted here what I see the role of non-equilibrium science for addressing large challenges. We do fundamental science at pore-scale fluid mechanics, and this gives us insights in the way that we can control reactions during forces or flow to generate diagnostics, or develop responsive and smart materials for anti-fouling applications, or create hierarchical functional structures. Okay. So science is so exciting. But again, because we're at a meeting that's interdisciplinary, I want to say that scientists have to work not only with each other together, but also with humanists, with social scientists, to start to think about the aspects of challenges that are beyond that can be addressed by technology alone. So gracias por su atención, and thank you very much for the invitation.
Well, thank you so much, Jacinta, for a wonderful talk. I think we have the time for at least 3 questions. Somebody in the audience? Rafael, could you please identify yourself and make the question?
Do you know of any structured channel that could be devised in a way that acts like the shark skin in order to improve or diminish the viscosity?
Yes, so actually one of the biggest challenges in terms of microfluidics is in thinking about how we go from laminar straight flow, set by the Reynolds number and the flow conditions, towards more turbulent flows that would increase. So the simplest way to do this was shown by George Whiteside's group at Harvard about 12 or 13 years ago, is to pattern microscale ridges on the channel. You can play this game. That seems to be the most effective way that I know of. You can also manipulate the geometry of the channel, and this allows you to create effectively turbulent motion.
I can use a combination of geometry and the contact angle or the wetting properties.
That I don't know of, but my knowledge is finite. So yes, and I will almost certainly guess that somebody has done this. And in particular, okay, so now in a microfluidic context, no, but controlling chemical properties and topography is one of the best routes towards creating what are known as superhydrophobic surfaces. And these mimic the natural lotus leaf. And usually people use a combination of physical structure along with chemical. I don't know of applications directly for flowing, but this is widely used as anti-oleophobic surfaces.
Somebody else?
Ramón Castañeda de la Universidad de Guanajuato. Jacinta, when the colloids flow, so you know that there are hydrodynamic forces, and they sometimes, or basically they form hydroclusters. When you have now attractive interactions like these sticky particles, so the attraction plus the hydrodynamic forces enlarge the cluster formation?
No, actually, so it's known from bulk rheological studies that adding attractions will actually suppress hydrocluster formation. And I think the mechanism there is likely that, so let me try and translate this very quickly. So when particles are flowing, let me go back, since it's right there. Oh, no, it's not. Okay, so when particles are flowing very closely, there's a fluid layer between the particles, and squeezing out that fluid layer will cause the particles to stick together. And so if you've ever tried to pick up, for example, a coin that's submerged in water off the table, you'll feel that it pulls back on you. Okay, this is the basic idea behind hydrocluster formation. So my understanding is that by adding attractions, you prevent the formation of the fluid bridges that leads to hydrocluster formation. And this has been showed in detailed experiments, I would say best by the group of, now I've just blanked on his name, at Chicago, whose name I'm totally forgetting, Heinrich Egger. I would say is the best. And so there is a competition. We haven't probed in detail in our experiments, partly because there was this wonderful set of work that appeared about right when we were doing this. But there is a competition between hydrodynamics on the one hand and attractions on the other that have a more complicated interplay than what I've shown here.
Thank you. Jacinta, on behalf of the students in the audience, which will you have talked a lot about soft matter physics in order to get here. Which are other ways for a student considering what do I study, what do I specialize in to have a participation in
okay, so I have an odd answer
now
to this.
your presentation
okay.
on if you need it.
That's fine. I don't have what I need on there. So the answer is you study what you want that leads you towards your passion. Okay. So that's a cop-out, but it's not. My undergraduate degree is in mathematics. My graduate degree is in physics. And I decided I wanted to go to think about material interactions. So I went to go work for an engineer as a postdoc in material science. And now I am a professor of chemical engineering and petroleum engineering. Okay, so, and that's either funny or crazy, depending on how you find that. So what I would say, I would find a path that you enjoy, okay, and that you don't let that, for education. So broadly, education in the sciences can help you solve problems if that's your focus. I will put in my plug for physics, but that's just because I like physics. But there's amazing work done in chemistry and in biology. And increasingly, there's amazing work done at the interfaces. And so for students, I think I would encourage you to take a path that keeps you at the boundaries. And that might be the best way of thinking about how to connect science to translational applications. Right? Move within disciplines and move within fields. Get yourself exposed to new types of problems and new types of approaches. And so that's probably what I would suggest at that point. Do something you like and then go do something else.
Okay, we have time for one. Okay, we have the time for one last question.
Jacinta, do you have some slight tips, idea about how strong the small animal can be to move in between the grains and the...
Yeah, so let me try and think about how to translate that. So people have measured the adhesion forces of bacteria on surfaces. It depends on the mechanism that they use to stick. Bad physicist. I can't think. Okay. So the forces are on the pico, which is 10 to the minus 12 to nano, 10 to the minus 9 newtons. But what I'm failing at is to try to convert that into something more useful for anyone else. Physicists, help. So the forces are relatively weak. But they're still strong enough compared to the forces that are applied, for example, during flow, which are of similar orders of magnitude. But it depends on the mechanism. One of the reasons that bacterial adhesion is such a sticky, difficult problem is that there are so many ways that bacteria can stick, not just by the mechanisms I've identified, but by several others. And so anti-fouling strategies have to think about targeting all of them. But that's the order of magnitude.
I know I said that it was the last question, but it won't be. The last question is here.
Now the last question actually is related to your last comment. Usually the bacteria that are stick to the surface, there is a complex population of bacteria, right? So when you have you studied a complex population of bacteria that stick to these?
No, and I think this is one of the most interesting aspects that people can do right now. So all of the systems that we've worked with are clonal. They're genetically identical cells. But in real communities, as you correctly point out, it's not just one kind of bacteria. And indeed, different strains of bacteria can interact, both beneficially as well as antagonistically. So we've wanted to do these. I'm not a microbiologist, so I've been looking for a good collaborator who has interests in species competition.
You have one.
But no, this is a fascinating question and really has not been explored from the physical standpoint, but it's something that, again, would be wonderful to do. But really, and this is where I'll again say, when you work in hard problems, working with people who have contrasting expertise is so valuable as well. So I can do simple experiments, and we do some other, we have some collaborations, but more focused on the engineering side. But I would love to find microbiology collaborators. I think it's a great project for biologists and physicists together.
Well, I was also to say that Jacinta has put together both the aspects we mentioned in our panels, physics on physics, because she has talked about nanotechnology and also thermodynamics. She has put all that together. But now I realize that she has put all science and humanismo together with her last remarks. I think that that's exactly what this meeting is all about working together and finding new ideas. So thank you so much, Jacinta.
Thank you. Gracias.
Buenas tardes, les recordamos que de 2 a 3 es el lunch. Vamos a iniciar ya nuestro último día, las sesiones simultáneas de la tarde a las 3 en punto. Y después a las 5 tendremos nuestra última plenaria, que es del área de matemáticas. Los invitamos a todos a quedarse para después celebrar todos la clausura con un vino de honor. Muchas gracias.